So Denovo Neuroscience, our lead assay is a phase 3 ready assay. It's probably one of the first precision neuropsychiatry drug. It's a first-in-class triple reuptake inhibitor for biomarker-guided treatment-resistant depression. So for many of you probably know, depression is one of the largest indications in psychiatry. In depression alone, or MDD, in US, there are more than 31 drugs approved in MDD. However, 30% of those patients will become so-called treatment-resistant depression, or TRD. And this is really one of the major unmet medical needs because there are only 2 drugs ever being approved for TRD. One is a very old drug, Symbix from Lilly. It's not being used very often. There's only one new drug ever being approved in TRD over the last 15 years, that J&J's Spravato or esketamine. So, given this such a large patient population, it just essentially just one new drug. It certainly is not enough. So, depression drug development, the backbone is monoamine theory. Typically, the first-line treatment is SSRI, targeting serotonin transporter. And that cure about 30% of patients, let's say. The first drug didn't work. If you switch to SNRI, and that can probably cure another 30% of patients. And if you fail both of them, and you become TRD, that represents about 30 to 40% of all MDD population. So 20 years ago, and it's pretty logic, right? Why don't we develop a triple reuptake inhibitor targeting all 3 monoamines at the same time? It sounds great idea, and actually many pharma, including BMS, GSK, and several biotech, and all devoting into triple inhibitor. Unfortunately, till today, all of them failed, including the drug we're talking about today from BMS. So BMS, the drug called the liafensine, it's very potent, highly specific triple reuptake inhibitor. So BMS had run 14 clinical studies with this drug, 11 phase 1 and 3 phase 2b. So BMS actually built a large safety database with the drug. It's about 1,300 patients or individual being exposed to this drug. They had actually demonstrated probably one of the best safety profile in the whole class. Unfortunately, it didn't demonstrate the efficacy. So what I'm showing here is a 2 large phase 2b study aiming to demonstrate efficacy of liafensine in TRD patients. 006 study, the right line represents the control arm. The solid blue line represents the liafensine is a monotherapy treatment arm. And at week 6, you can see it's a small improvement in the liafensine arm, but clearly it's not statistically significant. In the second trial, 007 study, very similar trial design, same thing, the red line represents the control arm, while the solid blue line represents the liafensine arm. At week 6, where the primary endpoint and come from, there actually no separation at all. If you look at the sample size, there were roughly 200 patients per arm. It's a well-powered study. Given such a complete failure in both large-scale phase 2b study, the BMS really had no path forward. And they killed the program. Actually, they quit the CNS field all at once that time. So that's our starting point. Okay, why we're taking on a field drug like this? So the prime example on our strategy coming from oncology. So this is an eraser drug. 20 years ago, lung cancer drug, just like today's CNS drug, every phase 3 failed, including eraser. This is the first EGFR inhibitor. So you can tell the green line represents the eraser arm, the yellow line is the chemo control arm. So no separation, it completely failed. So AstraZeneca had to retrieve the drug from the market. By the way, the drug was approved by FDA based on pretty weak phase 2 data. Again, that's a major unmet medical need back then. So AstraZeneca, they tried to find biomarkers. They didn't. It's actually 2 academic labs identified EGFR mutation work as a biomarker for eraser. So this is a new phase 3 trial, and you can appreciate in the biomarker positive patient, the green curve, the survival curve, the higher the curve, the better the efficacy, which is opposite from MADRS score, right? and have much better efficacy compared to the purple, which is the chemo control arm. And you don't have to balance that, right? You can tell there's pretty substantial improvement. So based on this result, in biomarker, and was able to rescue a failed phase 3 drug, erase it back to the market, making it $1 billion drug. More importantly, saves tens of thousands of lung cancer patients' life. And moreover, since then, and lung cancer officially entered the precision medicine era. Now, lung cancer is no longer considered as one disease. It is divided into many different subtypes and highlighted by different biomarker. And then the different subtype will be treated with a different drug accordingly. So that's a prime example of precision medicine. However, the point we want to say here, all this drug, without a biomarker, every single one would have failed, just like eraser. Because if the drug only benefits 17% of patient or 7% of patient like elk, or sometimes even 1 to 2% of the population, there's no way you can run a success positive phase 3 trial without single out or enrich those responders. And we believe seeing SDD should be 10 times or perhaps 100 times more complicated than lung cancer, right? So if lung cancer is not one disease, how can seeing SDD to be one disease? So that's our belief. That's our model. We believe a lot of late-stage failure in CNS, they might still benefit a subset patient. If this is 1,000 patient, okay, even the drug benefit 200 right people responder, but the efficacy is diluted by the 800 non-responder. So that's our hypothesis. We think many of drugs fail because of this reason, right? And we acquire those kind of failed drugs. More importantly, the archived clinical sample for the patient who participate in those study. So the bottom black box is our platform. So we're not making a guess because we believe CNS today is still black box, right? Our knowledge is incomplete. It's sometimes even wrong. So we're taking a data-driven approach. We scan the whole genome, and we compare the genome between the right people and the gray people. Obviously, we're talking about the billions of data points, right? We use AI, machine learning, or different algorithm. So the goal is try to find, okay, what those 200 patient right people responder, they share in common, well, the 800 non-responder they don't have. It could be one gene, or it could be a set of this gene. In this case, there's a set of 3 genes can separate the right people from the gray people. So that's the biomarkers we're seeking for. All right, we're not stopped there. You see a lot of publication on similar approach. We need to do a new trial ourselves. In the new trial, we mostly gave the drug to the right responder. How do we know who are the right people? Using the biomarker we just discovered to pre-screen the patient. So everyone talking about the highest failure rate, right? You can imagine this trial most likely will be a positive study. Today, I'm talking about the program liafensine. We have completed all 3 steps. So this is a snapshot of a genome-wide screen. I don't want to bother you. So basically, you're seeing millions of SNP across the whole human genome from chromosome 1 all the way to chromosome 22. The y-axis here represents the association with the drug efficacy. So basically, you're looking for a peak here. You can appreciate at the chromosome 10, there's a single peak. At the top of the peak, that's the biomarker we're seeking for. This SNP, or single nucleotide polymorphism, located gene code anchoring 3. So that's our biomarker. By the way, that's a 2 years work, all right, just for that single map. Now we plot this biomarker data back to the MADRS score, all right. So the right line still represents the control arm. All right, now we divide the liafensine treatment arm into 3 subgroups based on the genotype each patient carries. So by the way, a little bit of information about this SNP. This SNP have 2 alleles. You have 1 allele from mother, 1 allele from father. Therefore, you have a 3 possible combination of genotype, AA, AG, or GD. So, okay, 66 patients carry the AA genotype, 74 patients carry the AG genotype. They were represented by the dotted blue line. You can tell easily their efficacy is about the same as the control arm showing the red line, right? However, for 46 patients who carry the GG genotype, their efficacy, the MADRS score, is substantially lower than the control arm, also lower than those patients who carry AAG. You don't have rebel stat, you can tell it's quite significant. We put the MADRS score number here, you can do the math. 4.2 MADRS score improvement. That is remarkable for TRD. just give you people who don't know the field, those 30 MDD drugs, antidepressant, average that 2-point improvement in depression. So we're talking a 4-point or higher improvement in TRD. That was remarkable. So therefore, we define GG as a biomarker positive patient and AAAG defined as a biomarker negative. So the other thing for whoever dealing with the big data, the other thing always a false positive, because when you scan millions of markers, so our p-value is like 10 to the minus 7. So that still means there are 1 out of 10 million, just by chance, you just find that biomarker. So we save some sample aside. Okay, those samples are untouched with the genome-wide scan. Only look at this single genotype. But we got essentially the same data. The GG genotype performed the best. That almost eliminates the chance of false positive. Okay, so, and we put all the samples together. Indeed, only 20% of the patients carry this biomarker. We call it anchoring 3 positive. And 80% patients carry the negative biomarker. You could tell, again, the patient who are biomarker positive have about a 4.7-point MADRS score improvement, while the patient who had the negative biomarker, there are no efficacy. Very much echo the cartoon I showed you at the very beginning. So that sort of explains why the drug failed, right? Obviously, we also look some other questions, and whether this biomarker is a generic biomarker for other antidepressants. From the BMS study, we also have a chance to look at those depression patients when they were treated with the duloxetine, which is a SNRI, a dual inhibitor. So you could appreciate there really not much correlation between the genotype and the duloxetine efficacy. Actually, as a matter of fact, GG genotype performed the worst. So 007 study, those patients, they were treated with either duloxetine or escitalopram, which is probably the most popular SSRI. And again, there's no separation among the 3 genotypes at all. So those results suggest this biomarker is not generic antidepressant biomarker. It's a rather specific biomarker for liafensine. Well, what is anchoring 3? Actually, it's one of the hottest genes in CNS over the last 10 years. It's a scaffold protein connect the cytoskeleton to the membrane protein, including ion channel and transporter, which happen to be the target of liafensine. So the anchoring 3 gene mostly expressed in the central and the peripheral nervous system. And there's a large number of publications demonstrating this gene plays a major role in neuronal cells development, mobility, proliferation, and signaling. So there's also additional animal model link this gene to TRD progression or mood stabilization. Moreover, one of the key reasons this gene is so hot is 10 years ago, there are several GWAS studies linked this gene with a few psychiatric disorders, including most notably it's bipolar and schizophrenia and depression. And one more piece of evidence coming from pharmacology. So in BMS study, they test this drug at 4 different doses, and there are no efficacy at any of the 4 doses. However, if we only zoom in to the 20% of patients who carry the biomarker, you begin to see a very nice dose response curve from 0.25 all the way to 1, then plateau from 1 to 2. This is actually the typical dose response curve for SSRI, because when you hit the receptor occupancy about 80%, you pretty much maximize efficacy. You add more dose, doesn't increase efficacy. Actually, on the contrary, you may actually get a little more adverse event. So that's also tell us those are the 2 doses we should test. Okay, now we're done with the retrospective finding. Now, from now on, it's all prospective study. So based on the data I just showed you, so our new global study, we're gonna test liafensine at 4 different doses, 1 milligram, 2 milligram. Knowing their efficacy is about similar, our primary endpoint is combining both doses together. The control arm is placebo. And we enrolled TRD patient and 6-week treatment, 4-week follow-up. The primary endpoint is week 6. This is a global study. We have a 38 US site, 4 Canada site, and 16 China site. Eventually, we enrolled about 200 biomarker positive patient and small number of biomarker negative patient. So here's the primary endpoint. At week 6, clearly we hit the p-value. So 0.0056. And you also can appreciate as early as week 1, we begin to see the separation, indicating the fast onset of this drug. So this is a breakdown. As I said, 4.4 MADRS score improvement, very similar to the 4.7 we have seen from BMS study. Obviously, we also look each dose by itself, just like we expected. 1 milligram and 2 milligram, their efficacy is about similar, 4.4 versus 4.5. More importantly, each dose by itself is statistically significant. Even the sample size was cut into half, from 123 dropped to 60. So this is a pretty robust, a positive TRD study. Just as you know, okay, outside psychedelic, it's really, really difficult to score any positive study in TRD, one of the hardest indications in psychiatry. Secondary endpoint, clinical global impression. This is CGI, both significant. Even the third secondary endpoint, Sheehan Disability Scale, which normally takes longer time to show the efficacy, at week 6, that's also significant. So we hit the primary endpoint and all 3 secondary endpoint. Very, very solid trial. We also put in a patient self-rating scale called PRDF. Again, so that's another angle to confirm the finding. They're all significant. Doesn't matter which angle you look at the data. Safety, as we expected, this drug is very safe. Indeed, the safety profile here is very, very safe. And nice thing about using placebo control, especially when you look at the headaches, 12% in the liafensine arm, but 14% in the placebo arm. Because a lot of so-called AE can be confused with the depression symptom itself. That's a good thing about using placebo as control. Yeah, there's some AEs is higher in the group, but it's pretty minor, especially consider those TRD patients. So this sort of high-level safety comparison. Obviously, we know Spravato had the dissociation, cognitive impairment, had black box warning. It's on the REMS program. We have 0 of those. And even Simvax have a wide range of safety issue. We have even 0 or just 1 star. And sometimes even placebo have 1 star. So has probably the best safety profile in the whole class. As a result, you can appreciate this is the first ever genetic biomarker guided psychiatric trial, a positive trial in TRD. So the result was published on JAMA Psychiatry a few months ago. If you're interested, I think it's freely available online. And you can ask me, I can send you the copy as well. Okay, we also look at the dropout rate. As I mentioned, efficacy between 1 milligram and 2 milligram is comparable, right? So the dropout rate, placebo 23%, 2 milligram 25%, about similar. However, 1 milligram arm only have 6.5% dropout. So that makes decision easier. So given the same efficacy, liafensine 1 milligram, that's probably the dose to go. So even last trial is a phase 2B, FDA had agreed, because that's almost 200 patients, that was considered a pivotal study. So you need 2 pivotal for NDA. So we already have 1 in the bag, just 1 more to go. The next phase 3 just essentially repeat the last phase 2B study. We'd actually just increase a little bit of power, so 300 patients will be 2 arm, because we're going to focus on 1 milligram. But we're probably going to have an open label that's for long-term safety, and that's all we need for. And you probably appreciate by now we create a new business model, how we're going to protect the patent, right? Because we're going to have a fresh 20-year patent life for the new biomarker. The reason we call it Denovo, because the biomarker discovery all novel. So therefore, you're going to have a new patent life. That's 20 years patent besides the 5-year NCE. So, and market, we already know the TRD is a huge market, right? We know Spravato already crossed $1 billion sale. J&J project going to hit $5 billion despite they have the generic competition. They have black box warning. So this drug, there's a picture, it's a white pill, once a day, 1 dose, and genetically guided. They're going to be exceeded $3 billion without even considering potential indication expansion. So this will be the first genetic test. We have market survey, and 76% of doctors say, yeah, they would like to order ever first genetic test. Actually, half the psychiatrists say they would like to expand the use of liafensine beyond TRD, even for MDD patient. So this is a 3 phase 3 trial for Spravato in their NDA package. So you can see their efficacy is about 3.2 to 4. So our first pivotal is 4.4, which is need to repeat that in a second phase 3 trial. And for fundraising, we need a $50 million to finish the next phase 3 short-term efficacy. We believe that will be enough to reach the next inflection point, probably going to trigger M&A. And for NDA, we need a long-term safety study and companion diagnosis. So $100 million going to carry us to NDA. We're going to actually spin this program and 3 other programs we acquired from Big Pharma. They're all late stage, failed phase 3, and 2 Alzheimer program and schizophrenia from Eli Lilly. They're all first-in-class drug, all small molecule oral pill. So those 3 are in the biomarker discovery stage. We're going to spin them into a new company called Denovo Neuroscience, because the other half of our pipeline is in oncology. So you don't see this go together in one small biotech. So Cooley is helping us. So this is the executive team. This is our scientific advisor board. Many of the names you probably recognize. Those are some of the brightest minds in the world, in the field. So, I think psychedelics going on at the same time. We're actually the front runner, even compared to the psychedelic. So we actually had the top line pivotal data from a year and a half ago. And Campus just report their first positive phase 3 just last year, only 3.6. We had 4.4. Remember, they're psychedelic. And the other 3 just finished phase 2. They're all psychedelic. We're the only non-psychedelic in this pack. And we're also a private company. And Wall Street really likes the stories. Here are some of the popular CNS analysts on the Wall Street. And hopefully, with a positive phase 3, we're seeking for potential M&A on this. I think that's the end of my presentation. Thank you.